Hi, everybody. We're now moving on to Chapter 12, Meiosis and Sexual Reproduction. I have posted your mitosis and meiosis questions for the module on Drop Send. There is a folder called Mitosis and Meiosis module. And I've had a few questions about due dates. The due dates aren't strict. They're more of a guideline for recommendations for keeping you on track so that you progress at a good pace through the remainder of the semester. I also recommend that you watch this brief 6 minute and 45-second video, which is an animation of meiosis. Certainly the animation helps you show the separations of the chromosomes better than what I can do on slides. All right, so there's certain pitfalls that come with understanding the process of meiosis. And of course, the difficulty always lies in differentiating mitosis from meiosis. So basically, cells divide and reproduce in two ways: mitosis, which is asexual, and meiosis, which is sexual. Mitosis results in two identical daughter cells, whereas meiosis results in 4 sex cells. So pay particular attention to the differences as well as the similarities as you advance through this material. As well, the concept of ploidy is always difficult for students to understand. And ploidy is the number of complete sets of chromosomes in a cell. So in a gamete that is haploid, that has one complete set of chromosomes, it would be 23 in a human. Whereas in a diploid human, it's going to be 2 complete sets of chromosomes in the cell, so you would have 46 chromosomes. Okay? So ploidy is the number of complete sets of chromosomes in the cell. Also keeping straight sister chromatids from the concept of homologous chromosomes, which we'll go over in a moment, and knowing the difference between a germ cell versus a gamete versus a zygote, and particularly the germ cell versus the gamete. The germ cell is diploid and is the precursor from which the gametes are made, and the gametes are haploid. 2 gametes will unite to form a zygote. Then we need to keep track of the concept of the centromere versus the kinetochore versus the centrosome. The centromere is a region of DNA which is highly constricted to form a tiny area holding the 2 sister chromatids together during cell division. Whereas the centrosome is an organelle which is made up of microtubules, and it nucleates all the microtubules inside a cell in order to form the spindle apparatus during prophase of cell division. The main difference between the centrosome and the centromere is that a centrosome is a cylindrical structure that forms the spindle apparatus by controlling the cell's microtubules, where the centromere is a DNA region that holds the sister chromatids together during cell division. Kinetochores are protein complexes assembled on the centromere of the chromosome. Spindle microtubules are bound to the kinetochores. And the main difference between the centromere and the kinetochore is that the centromere is the region where the region DNA where the 2 sister chromatids are held together after replication of the chromosome, whereas the kinetochore is the protein complex on the chromosome where the spindle fibers will be attached during cell division. Chiasma is going to be a point of contact between 2 homologous chromosomes rather than between 2 sister chromatids. And this is where you'll see an exchange of genetic material can occur between the homologous chromosomes. And this is where crossovers, when we get to that concept, occur. Chiasmata is the plural form of that word. A gene is a stretch of DNA or RNA that determines a certain trait, such as coat color in a dog. Alleles are the alternate forms of these genes, such as brown coat, black coat, orange coat. And we'll get into this a lot more when we get into genetics in the next chapter. And again in meiosis, when we get to the point of looking at the homologous chromosomes lining up and having crossovers, you'll hear the words bivalent and tetrads. And 4 sister chromatids aligned together are known as a tetrad. And the main difference between the bivalent and the tetrad is that the bivalent refers to the group of the 2 homologous chromosomes, whereas the tetrad is discussing all 4 sister chromatids inside of those 2 different homologous chromosome pairs. So when we studied mitosis, we discovered that we created basically 2 clones of the parental cell. This was asexual reproduction. So then why sex? Why did sexual reproduction evolve? Contrary to popular belief, it isn't just for recreation or pleasurable purposes, although I do predict we will have a post-coronavirus baby boom coming up, but that's a little aside note. The first eukaryotes to engage in sex were just single-celled protists that appeared approximately 2 billion years ago, and this is well before about the 1.3 billion years when we had the first animals with neurons capable of assessing pleasure. And asexual reproduction works really, really well when an organism is well adapted to its environment and the environment isn't changing and it just keeps reproducing and cloning itself, and you have a whole clone of organisms all living very copacetically in its non-changing environment. However, when the environment does change, say it heats up or some new toxin or something enters the environment, it can kill off a specific organism. And if it kills off that organism and all the other organisms there are clones, likely all of them will die. Sexual offspring arises when you have inherited traits from 2 different parents. And so you have constant recombination or mixing of new traits. The idea is through this recombination of new traits, we can improve the fitness of the offspring, and therefore we have survival of the fitness. Now, of course, not all new recombinations improve fitness, and the idea is that those will selectively not be passed on in successive generations. Therefore, in sexual reproduction, when offspring arise from 2 parents inherit genes from both, an adaptive trait can spread more quickly through the sexually reproducing population than through an asexually reproducing one. And collectively, the offspring of sexual reproducers have a better chance of surviving the effects of a harmful mutation. This is, of course, because all asexually reproducing clones would inherit a harmful mutation, whereas in a sexually reproducing group, only a certain number of sexual reproducers would inherit the harmful mutation. Thus, as a group, the sexual reproducers have a better chance of surviving harmful mutations or environmental changes than clones. And in your textbook, it talks about how one important source of environmental change is a shift in a community of interacting species, especially considering the interactions between a host and parasite species. And this is called the Red Queen hypothesis for the evolution of sex, which refers for a need of a species to evolve as fast as it can just to keep a pace of a co-evolving species. And this name comes from Alice in Wonderland, or Through the Looking Glass by Lewis Carroll, in which Alice must run as fast as she can just to stay in place. Increased allocation to sexual reproduction can evolve because of Red Queen interactions, only if the selection is strong enough to cause rapid switches in which gene combinations are favorable. And you can think of this now with coronavirus, will we start finding that the coronavirus is going to evolve really rapidly, facing the pressures from us trying to evade it and kill it and rid it from our population. And of course, when we think of sexual reproduction amongst human beings, we think of our own inherited traits. Did you get your mother's eyes and your father's nose? And you can see here on this next slide that I've done the genetic analysis through my family. And up there at the top, you can see my mom, Lois, my brother, Paul, and there's Mr. Supreme, Ian. And this chromosome browser tool allows us to compare and view DNA segments that are shared between my family members. So if we look at chromosome 1 and we compare the blue region on the chromosome to the blue color that you see is coordinated to that of my mother. And you can see that each of these segments are my family members as compared to my chromosome, which you don't see there. You can see that I share a large length or centimorgans of DNA with my mother, much more so than the red one in the center, which is comparing my chromosomes to my brother. And then the last underlying one is that teal color, which is how many chromosome lengths or how much DNA segment I share in common with Mr. Supreme. And of course, you can see that I share much more in common with my mother, which makes sense. I inherit 50% of my DNA from my mother. Now, my brother is a much closer relative to me than is my great nephew, Mr. Supreme. So in the red, I share a lot more DNA length of segments with my brother than I do with Mr. Supreme. Now, why do my brother and I not share the entire 50%? Well, the 50% he inherited from my mother is different than the 50% that I inherited from my mother. And I hope you start to understand that while each child will inherit 50% of their DNA from each parent, they don't inherit the exact same 50%. And that's how we get the recombination and the mixing. And you can spend the money if you want to and do the analysis with your family through a large number of DNA assessment organizations. I wouldn't recommend any of the ones that try to give you a health analysis or any of that, though. Okay, so we need to learn to keep track of our DNA content. We have lots of definitions to keep straight on this slide, and hopefully this slide will help you. The term ploidy refers to the number of sets of chromosomes in a cell. Most animal cells are diploid, containing 2 sets of chromosomes. So in diploid animal cells, which contain 2 sets of chromosome, each chromosome will have a homolog. And so up in that very first box next to the word diploid, you'll see an animal cell containing basically 1 chromosome homolog pair. Let's say a yellow chromosome that you inherited from mom and the blue chromosome that you inherited from dad. The chromosomes that carry the same genes are homologous chromosomes. Now, they aren't identical chromosomes in terms of you might have inherited the alleles or alternate forms of genes differently from mom and dad, say brown hair color from one and blonde hair color from another. So they are homologs in terms that they have the same genes, but they will have different alleles or different versions of those genes. I hope you understand that. Now we look at the box below that, where we have the arrows pointing to the 2 homologous chromosomes. But wait, look, they have been replicated. And this is what happens as you enter meiosis, just like we replicated our chromosomes before we entered mitosis. We still have our homologs or homologous chromosomes, which are the members of a given chromosome pair, where one member of the homologs is a chromosome from the mother, the yellow in this case, we're saying, and the other member is from the father, the blue one. But since we've undergone DNA synthesis, we have two identical DNA strands formed. Sister chromatids are formed from the same chromosome, and they are connected together at the centromere. And you will see that in meiosis, our DNA replication is going to be followed by two rounds of nuclear division called Meiosis I and Meiosis II that will first segregate our homologous chromosomes, and then we will separate our sister chromatids. And the result will form 4 haploid nuclei. Now let's look at haploid. So over on the right-hand side, we see a single chromosome. It is not paired with a homologous chromosome. So the ploidy is haploid. It has one full complement of DNA, or a single set of unpaired chromosomes. And this is what we will find we have in our gametes. And in the bottom box, you can recognize that you have one chromosome. It is haploid, but it has been replicated and it has 2 sister chromatids. So that is one chromosome. So it's N equals 1. It is haploid, but it has 2 DNA copies. And here we go again with another picture, just trying to get all this clear in our minds, of 2 homologous chromosomes. Again, keeping track of the 2 different colors. They look exactly the same. You see the banding patterns are the same, so they contain all of the same genes. But we could say the red chromosome was inherited from mom and the blue chromosome was inherited from dad. So while they carry the same genes, they carry different forms, alternate forms, or different alleles on those genes. So let's see what happens when we replicate these. And here would be replicated chromosomes. Here you see the 2 homologous chromosomes, and each homolog has been replicated to have a sister chromatid, getting ready for cell division. So you have 2 of the red and 2 of the blue. Now, just as we're talking about that, you can also see that we have a piece that was exchanged by recombination. We haven't talked about that yet. We'll get into that later in this lecture. So just kind of ignore that for now. But this is to help you keep track of the idea of what are the homologous chromosomes, the red and the blue, okay, one from mom, one from dad, and before we enter our cell divisions, we replicate our DNA. So we have 2 of each sister chromatid. And the sister chromatid is identical, is an identical copy to the original one. Okay? All right. Here is another picture from your textbook in which in the top on A, you can see we have immunofluorescence of the chromosome or chromosome painting. And this will show that you have the same order of the exact genes on each chromosome in the homologous pair. And in B below, a drawing of that fluoresced chromosome, you will see that they're trying to show you that while they have the same genes on each homolog, that they actually come in alternate forms, or they're different versions of the same gene. And hopefully this will start to become clear to you over time as we go through all of this. Okay, so the whole point of meiosis in animal cells is that we're going to have animal germ cells, which are diploid, and they will undergo rounds of meiosis, Meiosis I and Meiosis II, to give rise to haploid, eggs or female gametes, and sperm, the male gametes. Sexual reproduction requires different a different nuclear division mechanism than what we learned in mitosis, and this will be called meiosis, and it will have the chromosome number. And we need to have the chromosome number because through sex, we recombine two gametes to form one unique new zygote. So our germ cells start out with 23 chromosome pairs, or 46 chromosomes, and they will undergo meiosis I, in which they will have that chromosome number for the beginning of forming a gamete. So we will end up with a gamete that has 23 chromosomes, not 23 chromosome pairs. All right, so we start out just like we did in mitosis with interphase, where we replicate our DNA. And then in meiosis I, we will have what is called a reductional division. This is where we reduce the chromosome number from the diploid, 46 chromosomes or 23 chromosome pairs, to the haploid number, which is 23 chromosomes. Now remember, all 23 of those chromosomes will still have a sister chromatid. So then we have to undergo meiosis II, in which we will have an equational division just to separate the sister chromatids. Okay, but it's not changing the actual chromosome number, just the amount of the DNA replicated content. And this is where everyone has a hard time understanding this. Okay, so let's summarize. As we're going into meiosis, we first have to replicate all of our chromosomes, and so we form sister chromatids. We then enter meiosis I, which is going to partition the chromosomes of one diploid nucleus into 4 haploid nuclei. So instead of 23 chromosome pairs that enter meiosis I, we will come out with 4 cells that each have 23 chromosomes that are not paired. No longer are they homologous pairs. The chromosomes are separated into 4 nuclei. Then in meiosis II, we will separate each of the sister chromatids. Meiosis I involves 2 divisions of the genetic material. The first division is called the reductional division, or meiosis I, because it reduces the number of chromosomes from 43 chromosomes, or 23 chromosome pairs, to 1 chromosome. 1 of the homologs remains. Okay, so it's now just down to 23 chromosomes, or N. It started out as 46 chromosomes, or 2N. In meiosis II, the sister chromatids are pulled apart and becomes an individual unpaired chromosome. Chromosomes are then sorted into 4 nuclei, and this is what's called the equational division. And this is basically akin to what happens in mitosis. So let's look at all of this with pictures of the chromosomes themselves. So we started out with a cell that contained 23 pairs of homologous or matching chromosomes. One that was inherited from the person's mother, which we will see here as pink, and one that was inherited from the person's father, which we will look here at the picture as being in blue. Now, rather than representing all 23 chromosome pairs, we're going to look at 1 chromosome, what happens with only one of the chromosomes. And just to keep our terms straight here, when we're referring to a pair of homologous chromosomes or a homolog, we would say that the pink chromosome is homologous to the blue chromosome. They have the same genes, the same banding pattern, they just have different alleles or alternate forms of that gene. During interphase I, the cell duplicated its nuclear material. So each homolog was replicated, forming 2 identical sister chromatids. And that's what we see here at number 1. We have 2 homologs. The pink one is homologous to the blue. And each homolog had duplicated its nuclear material. So you see this as 2 sister chromatids for each homolog. At this point in time, there is the equivalent of 4 copies of each chromosome in the cell. Now, this slide is an overview, and we'll look at the more detailed steps in a minute. But step 2, we see that the homologs have been separated, and they will be packaged into new nuclei. And each nucleus is going to contain one of each chromosome pair or one of each homolog. So it is now haploid. Okay? We've separated the homologs and packaged it into 2 nuclei. So each nuclei contains 1 chromosome. Those chromosomes still have 2 sister chromatids, but they're 1 chromosome. This is meiosis I. And at number 3 is where we're entering meiosis II. And in meiosis II is where the sister chromatids will separate and are packaged into the 4 new nuclei. Each new nucleus has 1 of each chromosome, so it is haploid. And what is hard for students to understand is that when they look at number 2 and number 3, it is difficult to understand why both of those are haploid. And you have to remember that haploid is how many chromosomes you have, and you have 1 of each chromosome. However, in number 2, you have twice the DNA content because our chromosomes have been replicated. Okay? So those are sister chromatids, not individual new chromosomes. In meiosis, the diploid chromosome number is restored when the gametes that were formed will be fertilized, or they fuse into forming a zygote. Okay? And you can see why this is necessary. Without meiosis, if we fuse 2 diploid cells together, then we would end up with double our DNA content, and it would keep doubling with every generation. And we need to keep that same chromosome number in every new individual cell. Thus, the goal of meiosis is to form 2 haploid gametes, which can then fuse to form a diploid zygote at fertilization. And here you see that represented in a picture. We start out with the male germ cell, which is diploid, and it has to become a haploid gamete. Similarly, the female germ cell in the bottom also has to become a haploid gamete. So the egg and the sperm, the egg being the female gamete, the sperm being the male gamete, can unite through fertilization and form a diploid gamete. You can see if we fused the germ cells together, we would end up with too much DNA in the new cell. Okay, so now we're going to begin a visual tour of the process of meiosis. And I tried to go through and put everything for meiosis I in teal and everything in meiosis II in a darker blue so that we can keep the processes straight. We already went through mitosis, where most cells reproduce to form identical copies or daughter cells for the purpose of growth or repair. But parental gametes, the sperm cells and the egg cells, are unique from all those other cells in that they reproduce through the process called meiosis. And the purpose of meiosis is to shuffle genetic information and cut the cellular chromosome number in half from 46 chromosomes to 23 chromosomes. And in this way, when an egg and a sperm cell combine during fertilization, the resulting embryo will inherit the appropriate amount of unique genetic information from each parent. If sperm and egg cells were to divide and keep the same number of chromosomes that other cells do, then the resulting embryo would have twice the number of normal chromosomes. For example, if a human egg and sperm cell each combined 46 chromosomes, the resulting embryo would contain 92 chromosomes, and this is a chromosome number that is not compatible with the normal development of a human being and its survival. The process of meiosis will involve two cell divisions of the genetic material. The first division, again, is Meiosis I, which is the reductional division. And it's called the reductional division because it reduces the number of chromosomes from 46 chromosomes, or 2n, to 23 chromosomes, or n, where n describes a single chromosome set, the ploidy. In humans, the reduction division results in two sperm cells, each with 23 chromosomes, or an egg cell with 23 chromosomes, and one that ultimately ends up being discarded as what's called a polar body. But that's getting a little bit ahead. The second division is Meiosis II, and that's called an equational division. The two sperm cells will split again and produce four sperm cells, each with 23 chromosomes, or N. We use the same names for the phases of divisions: prophase, metaphase, anaphase, and telophase. And I hope this doesn't confuse you too much. We keep it straight by calling it prophase 1 for those divisions or those events occurring in Meiosis I, and we'll call it prophase 2, for example, for those events that occur in Meiosis II. So let's start with prophase 1. In prophase 1, our homologous chromosomes will condense, they will pair, and swap some segments. Spindle microtubules will attach to the condensed chromosomes as the nuclear envelope breaks up. In metaphase 1, our homologous chromosome pairs will align between spindle poles. The spindle microtubules will attach to each pair. So this should look semi-familiar. Here is a picture of what's happening in Meiosis I. In early prophase 1, the chromosomes become shorter and thicker and are more easily stained. They can now be viewed underneath a light microscope. The sister chromatids, which have already been replicated, and you see here, they are joined at the centromere. The nuclear envelope starts to disappear. Later on in prophase, the homologous chromosomes, the maternal chromosome 1, say in pink, and the paternal chromosome 1 in blue, will line up next to each other in a process known as synapsis. An aggregate of RNA and protein will hold these paired chromosomes together. The homologous chromosomes will switch a little bit of material in a process of recombination, which is called crossing over. Here, the two chromatids at the chiasma will exchange portions of DNA. In this way, some of the genes originally on the father's chromosome are now on the mother's chromosome, and vice versa. This shuffling process helps create and maintain variation in a species. It's another method by which we get new genetic recombination. And then we see also in late prophase 1, the nuclear envelope is beginning to disappear and the spindle fibers begin to form at the poles of the cell. Thus, now we're entering metaphase 1. In metaphase 1, the pairs migrate to the equatorial plane of the cell, which is along the middle there. You see it lining up just like you did in mitosis. The spindle fully forms and attaches to the centromeres. The pattern in which the homologs align during metaphase 1 will determine the combination of maternal and paternally derived chromosomes in the progeny cells. Each chromosome moves independently of all the other chromosomes, and this is where we have the phenomenon that we call independent assortment. And you'll want to remember that for as we get into the genetic chapters. Independent assortment. the randomness by which each chromosome ends up in a cell. For example, the copy of chromosome 1 that a gamete receives in no way influences if the same gamete will receive the maternally derived or paternally derived chromosome number 2. And now we'll move into anaphase 1 is marked by the separation of the homologous chromosomes. They'll begin to head toward the spindle poles. Okay, so remember, anaphase 1 is separation of the homologs, not separation of the sister chromatids. After anaphase 1, we will enter telophase 1. Here, a complete set of chromosomes will cluster at each end of the cell. The nuclear envelope will form around each set, and we will have 2 haploid nuclei. Let's examine this in pictures. During anaphase 1, the homologous chromosomes, each consisting of 2 sister chromatids held together by a centromere, will separate and begin to move to the opposite poles of the cell, pulled by the shrinking spindle fibers. Then we enter telophase 1. Here, the movement of the homologous chromosomes to the poles is completed. Remember that although there is one of each type of homologous chromosome at each pole, one homolog at each pole, there is a mixture of maternal and paternal chromosomes due to the recombination that occurred during prophase 1. The spindle will now disappear, and cytokinesis or cell division occurs, producing 2 haploid daughter cells. In each of the 2 haploid daughter cells, there will be only one set of chromosomes, 1N or haploid. However, the chromosomes are still in their replicated state and consist of sister chromatids, so there is twice the DNA content. The cells will then move back into interphase and mark the end of mitosis 1. Okay, so now we'll be starting the second part of meiosis, or meiosis II. Meiosis II is pretty much identical to mitosis, or somatic cell division, in which genetic material is transmitted equally, identically, and without recombination into the daughter cells. In contrast to a mitotic division, which yields 2 identical diploid daughter cells, the end result of meiosis is haploid daughter cells with chromosomal combinations that are different from those that were originally present in the parent. In sperm cells, we'll end up with 4 haploid gametes being produced, and in egg cells, meiosis will typically result in a single haploid gamete, a single egg cell, and the remainder of the genetic material becomes lost in the formation of what we call 3 non-viable polar bodies. And we'll get to that a little bit later. But basically all of the cytoplasm goes toward the one viable egg cell. All right, so before meiosis II starts, we'll have to undergo another interphase. Now, in contrast to regular cell division or mitosis, the second interphase takes less time. Here, the chromosomes are going to uncoil into very thin threads, and the nuclear envelope reappears. And most importantly, although proteins are manufactured during this interphase period, the genetic material is not replicated a second time. Okay, so that's different from the interphase that precedes mitosis. All right, so we start out with prophase 2, where the chromosomes are going to condense, and then the spindle microtubules will attach to each sister chromatid as the nuclear envelope breaks up. Then we enter metaphase 2, and metaphase is always marked by the alignment between the spindle poles. So let's look at this in pictures, because everything is always easier to understand with a picture. Okay, so in this first part, marked by number 5, is prophase 2. Prophase II marks the beginning of the second meiotic division. Meiosis II is similar in that each chromosome consists of two sister chromatids attached at the centromere. The goal of meiosis II is to separate the sister chromatids. In prophase II, the chromosomes again become condensed and the nuclear envelope begins to disintegrate. The chromosomes, which now consist of the two sister chromatids, begin to move to the equatorial plane of the cell. The spindle fibers once again begin to form at the poles. Now we start to enter metaphase II. During metaphase II, the chromosomes become aligned on the equatorial plane and the spindle fibers attach to the centromeres. After metaphase II, we enter anaphase II. Here the sister chromatids will separate and begin heading toward the spindle poles. Then, at telophase II, a complete set of chromosome clusters are at both ends of the cells. The nuclear envelope will form around each set so that 4 haploid nuclei will form, 4 sperm and the 1 egg plus 3 polar bodies. Let's look at anaphase II and telophase II. In anaphase II, the centromeres divide, effectively separating the sister chromatids, which move to the opposite poles as they are pulled by the spindle fibers. In telophase II, the movement of the chromosomes to the poles is completed and the spindle disappears. In spermatogenesis, the nuclear envelope will form around the 4 nuclei, which then separate into 4 individual spermatids. In oogenesis, the ultimate result will be 1 egg cell or 1 ovum and 3 polar bodies, which will be discarded. What we see here at the bottom lower right hand picture is the result of meiosis II is the formation of those 4 unique cells. Each carries a new assortment of genes and chromosomes, each with half the number of the original chromosomes. So in summary, meiosis is a form of cell division that results in half the number of chromosomes in sex cells or gametes, thus maintaining the chromosome number of a species from generation to generation. Additionally, genetic variability will result from the recombination, such as we saw in crossover, along with the independent assortment of genes of the genetic material. All right, let's go to some practice questions. Okay, let's just kind of review some of the genetic consequences of meiosis before we enter our practice questions. Remember, in meiosis I, there is a reduction of the chromosome number from diploid, 2 separate copies of each chromosome, to haploid, 1 copy of each chromosome, during the formation of gametes. There is a random assortment of the homologs, and we will have recombination through crossing over that results in additional variety in the genetic material that's passed out to each cell. Okay, so hopefully reminding you of those points will help you answer some of our practice questions. Which statement is false regarding sexual reproduction? Does it result in a new combination of genetic traits? Does it produce offspring that differ from one another and from the parents? Does it produce progeny that are clones? Or does genetic recombination in sexually reproducing organs confer a survival advantage when the environment changes? And of course, the false answer is that it produces progeny that are clones. That's what happens during mitosis, or binary fission for that matter. The purpose of meiosis is which of the following? The formation of genetically identical individuals, the formation of diploid gametes, cellular division producing 2 replica, each with an equal number of chromosomes in each resulting diploid cell, the formation of haploid gametes, or that haploid germ cells are formed. And of course, the purpose of meiosis is ultimately to form haploid gametes. And don't confuse that with germ cells. Germ cells are diploid, and they're the precursor cells that form the gametes. Sister chromatids are which of the following? Identical copies formed by the DNA replication of a chromosome, attached at the centriole, identical before and after crossing over occurs, or joined by a common chiasma. And sister chromatids are identical copies formed by the DNA replication of a chromosome. During meiosis I, the chromosome number is reduced from diploid to haploid. True or false? And yes, it is during meiosis I that we separate our homologous chromosomes and reduce from the diploid number to the haploid number. Okay, which stage is represented in this picture? Prophase 1 or 2, metaphase 1 or 2, and anaphase 1 or 2. Well, I hope you at least recognize that it is a metaphase. And it isn't metaphase 1 where we would see the homologous chromosome pairs being aligned between spindle poles. Instead, it is metaphase 2, okay, where we see the chromosomes aligned midway between the spindle poles. The spindle microtubules are attaching to the sister chromatids and are getting ready to move the sister chromatids to the opposite spindle poles, not moving apart homologous chromosomes. And in case that wasn't clear to you, look at this example. Which stage is represented here? Again, we see everything lined up midline, right, at the metaphase plate. But which metaphase is this one? This one is metaphase 1. And you see that what we're doing here is we're separating our homologous chromosome pairs that are aligned between the spindle poles. The spindle microtubules have attached the 2 chromosomes of each pair to opposite spindle poles. So go back and re-examine both of those pictures side by side and get that through your head so you can recognize that for a test. Here we have a fill-in-the-blank question. Sexual reproduction in animals starts with diploid, what kind of cells, that form haploid, another kind of cell? And the answer is that sexual reproduction in animals starts with diploid germ cells that form haploid gametes. Which statement best describes this picture? Are these 3 non-homologous chromosomes? Are they a haploid set of 3 chromosomes? Are they a haploid 3 sister chromatids? Or are they diploid 3 pairs of homologous chromosomes, or diploid 6 non-homologous chromosomes? And this is seeing if you really got that concept of haploid and diploid, and what are the homologs and what are the sister chromatids. And these are, of course, diploid 3 pairs of homologous chromosomes. Now here is a karyotype, and you have to tell me what ploidy is represented in this karyotype. Haploid, diploid, or triploid. Again, seeing if you got that concept. And I think you can see that here, triploidy is the result of an extra set of chromosomes. This can occur when 2 sperm fertilize 1 normal egg, or a diploid sperm, that had a strange event during meiosis, can fertilize a normal egg. And we'll go through some of this later when we get into the genetics chapters. But I want you to be able to recognize the difference of haploid, 1 full set of chromosomes, diploid, 2 full sets of chromosomes, and now you can see here triploid, 3 full sets of chromosomes. And of course, in a human being, this would not be compatible with life. Only very occasionally are babies with triploid genomes born, and they might live for a few hours or days. There's one baby that was reported in medical literature to live for about 10.5 months, but this is extremely rare. 2 out of 3 pregnancies will miscarry in the first trimester, and almost all other babies are stillborn. But you should be able to recognize on this karyotype of a fetus the triploidy, and that you have 69 chromosomes. It's a 69XXX triploidy is officially how we describe this. All right, let's move along. Homologous chromosomes become attached to either what in prophase 1? And the answer is the spindle pole. During prophase 2, what occurs? Does a new spindle form, crossovers, chromosomes are single-stranded, the cell is diploid, or the nuclear envelope reforms? And of these answers, during prophase 2, a new spindle fiber forms. What do you call different forms of the same gene? And the answer is alleles. And the definition of alleles are pairs of genes on a chromosome that determine the hereditary characteristics. An example of different forms or alleles of the same gene that I gave is the determination of hair color. So your father might have brown hair and your mother might have blonde hair. And these would be different alleles, but for the same gene, which is hair color. As we advance into our genetics chapters, you will see we'll examine Gregor Mendel's pea plants. We might look at the genes for plant height, and there could be alternate forms of alleles of these genes, say a dwarf plant for a short height and a tall allele for the tall pea pod plants. I'll do one more short lecture from chapter 12 about crossing over and chromosome segregation, and then we'll go on to our future chapters. So everybody, good luck, stay healthy.